This video is a short recap and introduction of our student project Physarum Polycephalum, a Lifesaver. The idea. If you are not familiar, Physarum Polycephalum is a protist slime mold, a single-cell organism that is able to connect its nutrient sources with the shortest path possible. With our project, we wanted to test this ability and also transfer it to emergency exit routes. When you face an emergency, how do you know which exit you have to take? This, or this one, or maybe this one? Well, in the majority of cases, you choose the one that is closest to you. In our project, we want to mimic this behavior with Physarum, using our faculty building as a model to prove existing exit routes to be the shortest path possible to leave the building. To accomplish this, we 3D printed each floor of the building and put the slime mold into different rooms, marking the exits with food. Cultivation. Our slime mold has specific needs that we have to meet in the lab for cultivating it. Naturally, it grows inside of forests on dead tree stumps or leaf piles, which is a moist environment. Therefore, we have to provide it with enough moisture to keep it from drying out. However, too much moisture can lead to mold forming. Under dead leaves, not too much light comes through, and subsequently, our slime mold prefers darkness and avoids light. Despite being a bit unusual in forests, oats are the slime mold's favorite food source. The perfect temperature for it to grow turns out to be 23 degrees Celsius. We received the slime mold in its dormant state, called sclerosia, on filtering paper. We placed it on sterile petri dishes with 1 or 2% agar that was autoclaved beforehand. Afterwards, we started to wet it carefully with sterile filtered water. Then oat flakes were provided to the slime mold as food source to wake it up. They were sterilized with dry heat before to prevent mold from growing in our petri dishes. Every 2 days, the oats were changed and more moisture in form of sterile filtered water was provided if necessary. The dishes were stored in the dark at 23 degrees Celsius. 3D printing. At first, it was important to know if the slime mold tolerates the material that was used to 3D print. For this purpose, some of the scaffold structures of the 3D print were embedded in agar. After cooling, the slime mold was placed on the agar together with oats. The results indicate that the material did not hinder the slime from growing. However, this plate did develop mold after a few days. Here you can see the process of creating the different floors of our building in CAD software. And afterwards printing it with clear resin in 3D. This is our slime mold in one of the floor models. For the video, a little unfavorable conditions were used. We needed light exposure. Additionally, we could not use a lid to prevent reflections, which caused the agar to dry out over the course of this recording. Therefore, the slime mold did not grow very much.